Welcome to Chapter 18 of Microbiology. This chapter discusses a variety of infectious agents that are gram-positive or gram-negative coccus. And so these you're going to see some culprits that you have encountered probably in the clinic, you've heard about before. But first, let's figure out how we grow them in the lab. The blood agar plate is actually 5% sheep blood on a tryptase soy agar. And so the sheep blood is necessary for a certain bacteria to be distinguished, particularly those bacteria that make hemolysins. So hemolysins are exotoxins that are specific for red blood cells. And so this is a non-selective media, meaning that any bacteria could grow on the media, but only certain bacteria are going to be considered differential. And they're differential due to the fact that they secrete hemolysins and will lyse the blood. And as a consequence of that, you'll get three potential outcomes. You will either have an alpha hemolytic bacteria, which will partially lyse the red blood cells, causing an olive green color. You will have beta hemolytic bacteria that will completely and fully lyse the red blood cells, resulting in a clear zone around the colonies of bacteria. Or you will have gamma hemolytic bacteria, meaning that there's absolutely no lysis, no change to the red blood cells in this blood agar plate, and so there's no lysis whatsoever. And so the exotoxin hemolysin is the specific toxin that does this process. The process, however, is called hemolysis. So the hemolytic activity broken down with notes shown here. So alpha hemolytic is that green coloration. Beta hemolytic is full hemolysis. Gamma hemolytic, as a reminder, is no hemolysis. So there's no change in color, no zone of clearing around the bacteria. So you can see the olive green brown coloring here of alpha hemolysis. Gamma hemolysis, where you have just bacteria growing on the plate. But then here, you have beta hemolysis. If you were to hold your plate up, you would actually see a complete zone of clearing, and you could see light passing through the plate. Beta hemolytic bacteria that we're going to discuss in this chapter are Staphylococcus aureus and Streptococcus pyogenes. So more often than not, if you go to a lab and you have a severe sore throat, they'll often swab your throat, and they're looking for Streptococcus pyogenes. They will do a rapid strep test to determine whether or not you have Streptococcus pyogenes, but they'll also grow this bacteria on a blood agar plate to look for beta hemolytic strains. So both of these bacteria possess exotoxins that cause beta hemolytic responses on a blood agar plate. So Staphylococcus. Staphylococcus in general are common inhabitants of the skin, our mucous membranes. They're normal commensals of our human flora. They often, when stained by gram stain techniques, they are this very grape-like clusters. And so the species that we'll be discussing in this lecture are Staphylococcus aureus, Staphylococcus epidermis, and Staphylococcus saprophyticus. Staphylococcus aureus, shown here, is actually a full colonization on the body. So you can see that in a normal person, you have about 27% of Staphylococcus aureus in the nares. 10 to 20% will colonize the respiratory tract. You have it all across the skin, axilla, forearms, hands. So Staphylococcus can be isolated from pretty much about all bodily parts. In some cases, Staphylococcus aureus, you have individuals who are considered carriers of this microbe. These carriers have nearly 100% colonization in the nares, and so nearly a third of the population are considered carriers of Staphylococcus. It is indeed still a normal flora, and so not causing damage or harm. But for clinicians, it's sometimes worrisome in the sense that there are methicillin-resistant forms of Staphylococcus aureus, and so as a clinician you may have to go through a nares swab and may undergo treatment if they fear that you have a MRSA strain. This bacteria is a mesophile. So mesophile is a bacteria that can grow readily at body temperature, which is 37 degrees Celsius. It is a facultative anaerobe. So facultative means that it is not always an anaerobe. Typically it's an aerobic bacteria, but it can grow in anaerobic conditions, which is how it's so readily available to colonize our entire body. Additionally, this bacteria can withstand high salt, right? So it is osmotolerant. Extremes in pH, so it can withstand things such as the acidic environment of the vaginal tract, as well as high temperatures. So it is a risk of contamination in the clinic since it is all over our body. The presence of this microbe can result in diseases, especially if it is a pathogenic form. There are quite a few virulence factors of Staphylococcus aureus. Staphylococcus aureus possesses quite a few enzymes, in particular coagulase. Coagulase coagulates plasma and blood, and it is actually common to most Staphylococcus aureus isolates, so 97% of those. And the reason that Staphylococcus aureus will possess coagulase is it blocks the blood, or the entry of blood, from clearing out this microbe. So if you have a staphylococcal infection, it will coagulate around the site of infection, preventing the entry of white blood cells. Staphylococcus aureus also produces DNase. DNase is necessary to digest free DNA. If this microbe is colonizing an area and there is a lysis of blood cells, you will find that that lysis of blood cells would cause a release of DNA into the blood, and the release of DNA circulating through the blood would actually act as an alert for the immune system, because DNA is normally localized to the nucleus. So free-floating DNA would be a big red alert for our immune system, and as a consequence, S. aureus is able to digest that to prevent that big red alert. Staphylococcus aureus is often not treated well by penicillins because it produces the enzyme penicillinase, which inactivates penicillin proteins. And so when we have staphylococcal infections, we typically go to other lines of treatment besides penicillin. Additionally, Staphylococcus aureus produces a variety of toxins, such as various hemolysins. They lyse various red blood cells depending on their type. Staphylococcus aureus also produces leukocidins. As you may have recalled from chapter 13, leukocidins are necessary for lysing white blood cells. In particular, shown here, it's that of neutrophils and macrophages. Both of these cells are highly phagocytic and important second lines of defense to remove bacterial infections. Staphylococcus aureus can also cause the secretion of enterotoxins, entero meaning targeting enterocytes. So these are going to induce gastrointestinal distress, especially if you have a foodborne infection from Staphylococcus aureus. It also produces a toxic shock syndrome toxin. Toxic shock syndrome toxin, you may have encountered this if you were a female. There is a pamphlet inside most tampon boxes now that suggests that tampons should be changed at a regular basis because of Staphylococcus aureus. The buildup of the toxin from Staphylococcus aureus can cause severe fever, vomiting, shock, and systemic organ damage if left untreated. Exfoliatin toxin. So Staphylococcus aureus secretes this exfoliatin toxin, and my brain always goes to, oh, let's exfoliate. Well, this is exfoliation in the really improper degree. It causes a complete separation of the epidermis, and it's quite dangerous in that of children or babies if they are colonized with Staphylococcus aureus after birth. So the toxigenic diseases that you should consider are that of food poisoning. Food poisoning is due to the stable enterotoxins resulting in GI distress, vomiting, and diarrhea. And so the enterotoxins, when they are in the stomach or gastrointestinal tract, you have the release of those toxins after colonization, which then causes the damage. Staphylococcal scalded skin syndrome. You can see here a couple images where it's on an adult. This is a complete staphylococcal skin syndrome of a baby. So you can see the red rash, you can see the desquamination of the superficial layers of the epithelia. And so this is caused by the exfoliatin toxin. Toxic shock syndrome. So toxic shock syndrome, signs and symptoms are shown here. You can see this red rash. This patient may be suffering from toxemia. 50% of all cases are indeed associated with tampons, hence the warning on the inner pamphlet of the tampon boxes. And it's because Staphylococcus aureus, even though it's always present in our body, if it reaches high concentrations that are abnormal, then the toxic shock syndrome toxin will be released and be present. Methicillin-resistant Staphylococcus aureus. So MRSA infections are on the rise, and it's due to the various poor prescribing mechanisms that we have done with our antibiotics. So more often than not, patients come into the clinic and insist an antibiotic be prescribed for various viral infections, and we've kind of changed to a consumer-driven system in the sense that patient satisfaction and happiness is kind of become more than that of patient care. And so the prevalence of these antibiotic-resistant strains of bacteria are just, skyrocketing. And the quantities of antibiotics that we have to treat infections are going down because we're continually using the medications that we have improperly. So methicillin-resistant Staphylococcus aureus is multi-drug resistant. It's not just resistant to methicillin. It does tend to cause skin or wound infections. Nearly 2 in 100 people are carriers of MRSA in their nares, which is really quite frightening. And so I've had students actually who have lost friends. So there was a young man that I had in class a few years back, and he had a friend that he played football with in high school, and literally, that weekend on Friday he was playing football. On Monday he had passed away due to a MRSA infection. And so it is really quite alarming and frightening. So MRSA is not just hospital acquired, it can be community acquired, as in the case with this young person, but hospital acquired are more common. So nosocomial acquired can lead really quickly to bacteremia and potentially septicemia. And so when you have bacteremia, it's, indeed the spread of, this particular microbe from the skin, contaminating other medical devices and then reaching the bloodstream. And so once it reaches the bloodstream, you're at risk for endocarditis. There's other illnesses such as necrotizing pneumonia, there's surgical site infection issues. Of course, now we know that there is vancomycin-resistant Staphylococcus aureus, so VRSA. And these are just some of the images of staphylococcal infections that are really quite severe. So how do these sorts of antibiotic resistance take place? First and foremost, it is indeed the natural evolution of a bacteria. And so you have a bacterium who has picked up a resistance gene. This bacteria is selected for because of poor prescriptions and antibiotic usage. So essentially what's happened, you have a variety of bacteria, but then you have this one resistant bacteria, but he's held in check as long as you have a normal flora present. But then once you take antibiotics, all of the normal flora, the microbes that were actually keeping this resistant microbe in check are going to die off, and then now you have only the resistant microbes taking place. And so you have a really, really unfortunate situation where you have a severe infection. And then of course, because there is not very little competition, the rest of the susceptible bacteria dry off, and then you have the reemergence and complete repopulation of the resistant strains. And this takes place because of multiple reasons. Poor patient compliance, we take our medicine until we feel better. And so if you're not finishing a full round of dosings, you are putting yourself at a much greater risk of obtaining an antibiotic-resistant infection because you're wiping out your normal flora, but then you're not treating the infection fully, and you're putting yourself at great risk. There is indeed overprescribing. So as I had mentioned before, there are practitioners who provide antibiotics for any infection possible. They cover just in case. We should not be prescribing just in case antibiotics. There's few antibiotic sources. The rise of antibiotic resistant strains is just constantly going up. And so there's definitely a need for a revolution, in my opinion, in the medical system for this. There's, of course, improper disposal. So depending on how you get rid of your microbes, there's, of course, improper disposal depending on how you get rid of your drugs. Flushing them down the drain is not a proper approach. And then, of course, improper disposal actually goes to that of our industrial properties. So we have a lot of industrial farms that provide prophylaxis antibiotics to cows and chickens to increase the rates of growth, their size. And, the runoff from those farms and facilities in turn reach our water supplies and have, in some regards shown some links to, transmitting to patients or people within the community who are, exposed to those contaminated sources. And so the molecular mechanism of antibiotic resistance takes us back to the process of conjugation. So conjugation is when you have a bacteria that has indeed a plasmid. That plasmid then is transferred from one of the plasmid containing F positive bacteria to an F minus bacteria. And then of course, then you have two bacteria now that possess the resistance gene that may indeed be on the plasmid. And that resistance gene could be that of, methicillin, it could be that of other antibiotics, or even a whole host of antibiotics in the case of like NDM1. Other staphylococcal infections include that of Staphylococcus epidermis. Staphylococcus epidermis is a commensal in the body. It causes various opportunistic infections, and in some cases it's now been known to cause nosocomial infections. It's highly prevalent in that of catheter-related blood infections within the hospital setting. And so nearly $2 billion are expended annually due to this type of infection. And honestly, if we think of how these sorts of infections are taking place, as you clean the site to insert a catheter, you're actually in a highly bacteria-rich environment, and then you insert the catheter in, and so there's a much higher risk of entry of this particular microbe. And then, of course, with other indwelling medical devices or a heart cath, it's certainly a much more alarming illness. And then when we think of Staphylococcus saprophyticus, so epidermis is located all over the skin. Staphylococcus saprophyticus is located in the gastrointestinal tract. And since its location in the gastrointestinal tract is so close to that of the urethra, it leads the second leading cause of uncomplicated UTIs. It's right behind that of E. coli. And so it's essentially its main virulence factor, lipoteichoic acid, allows this bacteria to adhere to the urethra surfaces and colonize and cause infection. One of the major treatments that of a UTI is high quantities of fluids, water. We would suggest things like cranberry juice, which will release some of those adherence properties of bacteria. So groups of Streptococcus. So the various groups of Streptococcus are broken down into A, B, C, G, and D. We're going to discuss primarily A, B, and D. C and G are much less common. They tend to cause just acute infections such as those, and they're picked up more often from animal flora. So group A, Streptococcus, is that of Streptococcus pyogenes, the causative agent of strep throat. Streptococcus pyogenes can unfortunately lead to a very unpleasant skin condition such as emphysema, and so you can see emphysema here. It also, if it becomes invasive, can cause things like necrotizing fasciitis, shown in both of these images. Or, of course, it has its own version of a streptococcal toxic shock syndrome. And so, more often than not, this is a non-invasive microbe. It's an inhabitant of the nasopharynx, occasionally in the skin. But then, of course, it can cause severe disease. In the case of strep throat, the biggest concern is that of asphyxiation, where the throat wall is, the airway has closed off. So our next bit of info is to introduce you to some immunology terms so that you can understand some virulence factors that are possessed by Streptococcus pyogenes. So first of all, what is an antigen? I've shown here a prokaryote, and in this case, this is a gram-negative bacteria. I have enlarged the lipopolysaccharide on the cell surface and shown that antibodies can bind the structure of lipopolysaccharide. And that's exactly what happens with our immune system. So bacteria that are not meant to be in certain locations can be bound by antibodies. And so this lipopolysaccharide is considered an antigen. So it is a substance that is recognized by antibodies, and it's recognized as being non-self. And so these antibodies allow our immune system to mark something as foreign, And then when we have this object marked, this lipopolysaccharide, it then can be digested by phagocytes. So phagocytes will actually come. They have a specific cell surface receptor that is specific for this heavy chain on the antibody. It will bind to that heavy chain and then induce phagocytosis of the entire complex. Streptococcus pyogenes has distinct virulence factors or surface antigens that protect it against our immune system. One of those surface antigens is C carbohydrates, and C carbohydrates protect this bacteria from the lysosome. So the lysosome is that structure necessary to digest and break up microbes. And so Streptococcus pyogenes being able to survive the lysosome allows it to persist within our bodies. The fembrae that are necessary for attachment serve as a surface antigen that can be recognized by our immune cells, as well as M proteins, and I consider M proteins as like chomping down because they allow the resistance of phagocytosis. Additionally, there are things like hyaluronic acid capsule. My example for this is the invisibility cloak that was possessed by Harry Potter in those awesome series. So the hyaluronic acid capsule provokes no immune response, so it allows the bacteria to hide out within our body and is not recognized by the immune cells. There are extracellular toxins. In particular, there is streptolysins, which are distinct hemolysins possessed by Streptococcus pyogenes. These cause cell death and tissue injury, so they lyse our red blood cells. There's also erythrogenic toxin, which is a pyrogen, so pyro for fire. This induces fever and a typical red rash when it is released at the site of infection. Additionally, there are superantigens. So superantigens are distinct distractive antigens that are secreted by Streptococcus pyogenes, and they initiate a very, very strong immune response. And so instead of being recognized by the immune cells in a typical fashion, they actually are recognized by the immune cells by binding just only a portion of the immune receptors. And so any of the cells that are replicating, trying to get rid of superantigens, they're actually replicating and producing specificity to the wrong protein. And so as a consequence of this, the superantigen can cause tissue necrosis and quite a lot of damage within the host immune system. Long-term sequelae of Streptococcus pyogenes infections is that of rheumatic fever. So rheumatic fever is because of Streptococcus pyogenes actually moving from the original site of infection and causing carditis. So extensive damage to the heart. In particular, it can persist and cause things like arthritis or fevers. There's also acute glomerulonephritis, so damage to the kidney. And so persistent streptococcal infections can, in addition to causing heart failure, can cause kidney failure. Group B Streptococcus. This bacteria resides distinctly in the human vaginal tract, the pharynx, and the large intestine. It is of the greatest risk for neonatal pneumonia. So newborns within the first week of life, if they are infected with Group B Streptococcus, can be at higher risk for things such as sepsis or meningitis, so inflammation of the meninges. The illness is highly prevalent that in newborns or, of course, adults that are over 60 years, both of which are immunocompromised patients. Group D Streptococcus is the Enterococcus. It is typically a normal colonist of the normal GI tract. It is often non-pathogenic but causes opportunistic infections and nosocomial infections. So you can see here that catheters that are inserted can result in dissemination of the bacteria. Colonized patients then can result in contaminated equipment or fomites. It can contaminate hospital staff, so fomites on their sleeves. They can then become passive carriers or even asymptomatic carriers to new susceptible patients. Or because it is a GI tract infection, it can contaminate our respiratory sources or potentially the sinks and then door handles, so fomites that then can contaminate the new patient. There are strains of Streptococcus that are vancomycin resistant, so vancomycin resistant Enterococcus, or VRE, is a prevalent problem in the clinic. Streptococcus pneumoniae is the pneumococcus. It causes nearly 60 to 70% of all bacterial pneumonias. In most cases, there are quite a few people that possess this normally in their nasopharynx, so they can spread to susceptible patients. As a consequence, this is typically community acquired, so passed from person to person. And so you can see the pneumococcus here. It has a distinctive capsule which protects it from phagocytosis. Neisseria gonorrhea. Neisseria gonorrhea is the gonococcus. So this is an STI infection. Nearly 820% of people in the United States have reported new cases annually. This bacteria replicates prevalently in the reproductive tract and urethra of men and women. The major virulence factors of consideration for this bacteria are fimbriae. So as I've mentioned before, if a bacteria cannot attach, it cannot infect. And then this other enzyme here, IgA protease, is a protease. It's a enzyme that will cleave secretory IgA. IgA is an antibody that is specific for the mucosal tracts of the respiratory tract, the reproductive tract, and so by cleaving this is actually removing the antibody that would fix this particular gonococcus during an infection. This bacteria is a gram-negative intracellular bacteria, so it in particular is often phagocytosed by neutrophils, and then because it is intracellular, it's able to persist in the neutrophil, causing infection. Neisseria meningitis causes the disease meningitis, so inflammation of the meninges. It is most commonly spread through the exchange of respiratory droplets, throat secretions. It is spread in places where there are high close counters, it is spread to high-risk individuals who are living in overpopulated areas with close quarters, especially those that are children or even young adults. The disease in this case often happens very rapidly, resulting in neurological symptoms. This particular bacteria possesses the endotoxin lipopolysaccharide, being a gram-negative bacteria, which can cause hemorrhage and shock. The virulence factors of this microbe are capsule, meaning it's resistant to phagocytosis, fimbriae, which are necessary for attachment, IgA protease, which are now lysing antibodies preventing their efficacy, and then of course the endotoxin lipopolysaccharide. Just as a bit of a refresher, if you've not had anatomy before, the meninges are the outer covering around the brain. So the meninges, inflammation of the meninges or dissemination of this microbe into the meninges is quite deadly. And so early signs and symptoms are that of cold hands and feet, complaining of limb pain, and then they progress pretty rapidly within 13 to 22 hours to rash, headache, stiff neck. Stiff neck is a high indicator of meningitis. And then you can see here the images of a rash. Prevention of all of these infections is that of good hygiene, such as hand washing, proper PPE, so wearing gloves, doing excellent techniques, having sterile technique in the case of surgeries, or at least aseptic technique when you're dealing with patients. The common cause of meningitis in young adults is considered which genus? Is it Streptococcus, Staphylococcus, Neisseria, or Bronchomula? So Neisseria meningitis.